Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about decalcification, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss rationale and aims, learning outcomes, indicative content, resources, submission of evidence, Q&A suggestions, some evidence ideas, reflective statement, and some helpful tips to make your portfolio better. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into this submodule. Rationale and aims of this submodule outline the general information and expected knowledge and competence that needs to be achieved when this submodule is completed. These are relevant to each submodule. For this submodule, the expectations are that the candidate will be able to handle various tissues containing calcium for histological processing, gain clear understanding of the principles and practice of decalcification and how it is successfully carried out in the lab, gain knowledge of different decalcifying reagents and their use with different tissue types, use and dispose decalcifying reagents safely. The learning outcomes demonstrate candidates' knowledge, skills, and competence in this submodule. They also include demonstration of the understanding of scientific principles, laboratory procedures, quality assurance, and professional conduct. They allow candidate to work autonomously and solve problems independently. Let's explore the first learning outcome, which is for the candidate to explain the principles of decalcification and the methods available. What is decalcification? What methods of decalcification are available? Consider principles of decalcification like ion exchange, chelation, and solubility in acid, and methods of decalcification like chelating agents, acid-based, electrolytic, surface and microwave-assisted decalcification. The second learning outcome is for the candidate to discuss when decalcifying agents should not be used and explain why. When should the decalcifying agents not be used and why? What would be their impact? Consider non-calcified tissue, samples for molecular studies, samples requiring sensitive IHC staining like HER2 or some special stains like Grocott, unfixed tissue, when assessment of fine cellular detail is required, samples that are very tiny, when calcification is a diagnostic indicator, or when detecting microcalcification. Possible consequences could be degradation of genetic material, or protein integrity, morphological distortion, staining interference, loss of antigenicity, loss of diagnostic indicators, incompatibility with further testing, and so on. The third learning outcome is for the candidate to describe how decalcification may affect subsequent methods of analysis. What are the effects of decalcification on subsequent analysis of tissue samples? Consider H&E staining quality due to reduced staining intensity of nuclei, affected molecular analysis due to damaged genetic material, prevented mineralization assessment due to loss of minerals, extended duration of decalcification can damage tissue, IHC staining affected by damage to antigens, and so on. The fourth learning outcome is for the candidate to demonstrate with examples the preparation, monitoring, and processing of specimens for decalcification, maintaining records and audit trails as appropriate. How would you prepare, monitor, and process samples for decalcification? How would you record decalcification process? How would you keep audit trail? Consider fixation, specimen trimming, selection of the appropriate type of decalcifier, using temperature and agitation to speed up the process, monitoring using physical test, endpoint testing via chemical or radiographic tests, recording regular checks and endpoint test results, documentation, sample washing following decal and further processing of decalcified sample. The fifth learning outcome is for the candidate to demonstrate with examples from practice decalcification procedures on a variety of calcified tissues and explain how to test for completion of decalcification. What is the decalcification procedure used in your laboratory? Is it the same procedure for all samples or do you have different procedures for samples? If so, why do they differ? What types of samples are usually decalcified? How would you test for completion of decalcification? Consider different decalcifiers and decalcification methods for different samples like bone marrow trephine, femoral head, and other sample types. Consider time point tests like chemical, physical, bubble, weight loss, weight gain, or X-ray. The sixth learning outcome is for the candidate to discuss the risks and hazards associated with the use and disposal of decalcifying reagents. What risks and hazards are associated with decalcification? How would you safely use and dispose of decalcifying agents? Consider HNS risks like corrosive burns, respiratory hazards, acute toxicity, toxic fumes, or sensitization. Consider quality risks like over-decalcification, morphology damage, molecular alteration or equipment damage, COSH, SOPs, PPE, risk assessments, training, regulatory bodies, waste segregation. Consider disposal risks like contamination, environmental toxicity, dangerous reactions, or presence of hazardous materials that require licensed contactors to dispose. The seventh learning outcome is for the candidate to demonstrate with examples from practice how to troubleshoot problems which may arise, including root cause analysis as appropriate. What are the common problems associated with decalcification? How would you troubleshoot them? What could be their consequences if not resolved? Consider issues like pore fixation, concentration and type of decalcifier, duration of decalcification, tissue size, reliability of endpoint testing, irregular monitoring, tissue damage, and so on. Also consider troubleshooting approach like problem identification, data gathering, root cause analysis, corrective and preventive action, communication, monitoring, and documentation. The eighth learning outcome is for the candidate to explain the relative merits of decalcifying agents used within cellular pathology and be able to select the appropriate reagent for a range of samples. What reagents can be used as decalcifiers? When would you use each? What is their speed of action? What is their tissue evasion ability? What are their effects on subsequent analysis? Consider different types of decalcifiers like strong and weak acids, chelating agents, ion exchange resins, and so on. Also consider their properties, mode of action, pros and cons, suitability and compatibility, available alternatives. Those are the 8 learning outcomes that have to be met for this submodule. Indicative content highlights knowledge and understanding requirements that might be assessed during the Viva. They may or may not be demonstrated by the learning outcomes or candidate's evidence. In the decalcification submodule, candidate is expected to have knowledge and understanding relevant to homeostatic regulation of calcium levels. How does body regulate calcium? Consider parathyroid hormone, calcitonin, vitamin D, feedback loop. Pathological significance of calcium in tissue. What could be the pathological significance of calcium in tissue? Consider dystrophic and metastatic calcification, organ or tissue dysfunction, malignancy marker, cardiovascular disease, inflammation or cell death, neurological disorders, musculoskeletal problems. Resources section on the Brightspace contains some very useful information. Submission of evidence on Brightspace informs the candidate what evidence needs to be submitted for each subsection. Candidate needs to submit answers to questions that were set by the training officer. I will share some examples of possible questions later on. Candidate also needs to select an additional piece of evidence relevant to this subsection. I will also share with you some ideas of possible evidence later on in this video. Each submodule requires the candidate to submit their reflective statement relevant to their practice and training in each submodule. Questions should be set by the training officer. They need to be based around the knowledge requirements, relevant for each subsection. They should allow the candidate to demonstrate their understanding in each subsection. The questions should be specific and relevant to the candidate's in-house practice. They should also allow the candidate to demonstrate their specialist knowledge. If you would like to see specific example questions, please have a look at the video for trainers. Link will be included in the video description. These are some ideas you could explore for your chosen piece of evidence. And some more ideas for your chosen piece of evidence. Each submodule also contains reflective statement which is asking you, the candidate, to summarize your role in the laboratory in the context with each submodule. How do you demonstrate that you can draw conclusion from what you have learned, reflect on your current practice and how it can change in the future due to gained knowledge? Think about what is your laboratory in context this submodule. What impacts can your actions have on your colleagues, department as a whole and for the patients? You also need to write a personal reflection on your training and why you chose your evidence. This is very personal to your own circumstance. It should demonstrate your ability to be self-aware when analyzing the gaps in knowledge. You can use it as an opportunity to reflect on aspects of your training, application of your gained knowledge and skills, and how you have achieved your goals. Think about what your evidence demonstrates. Why did you choose this specific evidence for this submodule? Also write what your training was like when it comes to this submodule and how your practice changed due to your training. The reflective statements can also be used as a CPD evidence in case you are chosen to be audited by the HCPC. I would also like to share with you some helpful tips that can make your portfolio look really good. Try to present the information in various formats such as tables, pictures, photographs, diagrams, flowcharts, graphs or even videos, if you can. This nicely breaks down a lot of text that is usually present in specialist portfolios. Try to make it a little bit more interesting to the reader, which will be your training officer and eventually the assessor. Try to put yourself in their shoes. How would you like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture is worth 1,000 words. I will make a video. I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also try to use various types of evidence, not just one type, like case studies, for each submodule. You could use experiments, case studies, annotated results, annotated photographs of you performing tasks, evidence of troubleshooting, annotated EQA results, nonconformances, validation and verification evidence, comparisons of different approaches and their pros and cons, control material production, certificates of course attendance with reflective statements, annotated images or charts, and so on. Try to bring in your personality. Think about the ways you like to learn best. If you like experimenting, perform experiment. If you like troubleshooting, perform troubleshooting. Choose something that demonstrates your understanding, knowledge, skills, and how you apply this in your practice. And one more thing, keep the learning outcomes in mind when you selecting your evidence. Good evidence will tick off several learning outcomes. And this brings me to the end of today's video. Hopefully you now have a better understanding about the learning outcomes for this submodule. And I hope you also have a better idea what evidence you can choose to demonstrate your competence. And my final take-home message? Keep those learning outcomes in mind when answering questions and selecting your evidence. Bye for now.